Well, hey there, Biology 11. So this is bonus topic number 2, stems and leaves. There's a lot to talk about with stems, different modifications. We've already talked about xylem and phloem. There's a little bit more that we kind of got into, a little bit about the rings and about the cambium and topics like that. Plus, we can't leave out the fact that we still need to look at leaves and photosynthesis, and there's some pretty interesting modified leaves out there. So let's get into this topic, shall we? Here is sort of like the great clock of the history of our planet. And what I find really interesting about this, the reason why I wanted to show you this, the first life forms, if you think about it in terms of pretty well one day, the first life forms show up around 4:30 in the afternoon. Okay, it takes quite a while. Those are bacteria, and it isn't until really around 9:30 at night that plants invade land. Angiosperms show up. They kind of crash the party at about 10:50 at night there, if I had to call it. And this abrupt little species called Homo sapiens door crashes at about 11:50. That is a pretty interesting context for thinking about life on the planet. You've seen it a couple times in the course, but I think it's well worth watching. So there's our dicots. Uh, we look at flowers in patterns of 4 or 5. Pretty interesting when you actually take a look. It's like, really, that cactus has a taproot? Yeah, it's a dicot. Not all are woody species. Remember, they don't always have to lay down um, heavily reinforced secondary xylem. They don't have to do that. Some do, some don't. And then there's our grass-like plants. Rice, for example, one of the most important food crops on the planet. And if you look at it up close, you'll see that parallel venation, if you look carefully, I'll just draw a little bit. You can see that, like corn, it's got parallel veins in its leaves. Wow, imagine that. Look on the other side. There's a coconut palm tree there, and if you look at the fronds, they have parallel venation, right? And it's unbelievable to think that a coconut tree that big has fibrous roots. Well, now that you've seen the history of the sort of very primitive plants, you think back to the pterophytes and think of ferns, and now you look at a coconut palm, you can sort of see a degree of relation. Again, here's our monocot and dicot chart. I won't spend too much time with this. Suffice it to say that unless you arrange your xylem, there we go, towards the inside, if you don't put that xylem towards the middle and do that for all of your veins, you will not get the potential for rings of woody tissue. You just can't. So it would be in your interest to make sure that you know this chart. You've seen it before, it's been in other presentations. It differentiates the monocots from the dicots, and you really do need to know that. I'm just going to move on a little bit. This chart goes on and on. The reason behind this slide is pretty self-evident. Your vascular cambium, right here, is cells that can produce your xylem and your phloem. And that growth pattern determines whether or not you've got kind of a corn-like structure like grass plants, where they can be kind of rigid and reinforced. Bamboos has some pretty woody growth, but it's not. Even bamboo doesn't lay down ring after ring after ring of xylem in towards the center. They just don't do it. So this is a neat sort of slide. If you draw that ring through the cambium, you can see that this can grow its xylem in towards the center. And if it keeps that up, you'll get ring after ring every year. Sometimes a little bit hard to see, but there's your vascular cambium, your phloem, and your xylem. And the xylem's a little bit easier to note because it's got these large, hollow looking tubes. Okay, so there's your xylem tissue there. Right? Phloem is much, much finer than that. If you look at monocots, It sort of looks like an alien, doesn't it? I always thought it was funny. They Each one of these right here, um, these are your this is xylem. These are vessel elements, pretty big xylem. Corn's a very efficient transporter of water. There you go. But the phloem fibers are largely up here. They're much finer, and you'll see like little tracheids down here hanging out with their vessel element counterparts. But when you look at it, these could grow. You look at this one; he's facing in towards the middle, right? Some of them, um, some of them don't. Some face at different angles, and it's just not organized enough to set up a ring. It's not like everybody's lined up like this, right? And everybody's xylem grows into the middle and their phloem grows towards the outside. That doesn't happen. It's too scattered. And for that reason, sure, you'll get woody growth, but you won't get heavily reinforced woody growth. So, secondary growth, I kind of already spoken to this, right? So I'm going to move through that. There's a little bit of material I think that you would look at if you were taking a forestry course. Suffice it to say that your um, growth year after year is a result of laying down secondary xylem. Pretty neat when you get a chance to look at a woody stem in cross section like this. We've looked at that summerwood, right, which is this thinner section here, and we've looked at our springwood, which is much better, much more elaborate growth from here to here, as opposed to the summerwood. Um, if you wonder what happened to your primary xylem, it's down here getting crushed. It's getting destroyed, and it'll end up in the pith eventually, and then that's it. Crunch time. It's gone. The newer tissue is really all that's very functional. Um, the vascular cambium stays up pretty high. In fact, you see it grow and it migrates towards the top, laying down the newest xylem, right, and the newest phloem each year. Now, there was one called cork cambium, and I didn't want to get too distracted by it, but the cork cambium can lay down that new layer of cork sort of year after year. And cork itself is pretty protective, right? It's just under the bark. And you need to lay that down because as the plant grows in girth, you've got to have something to keep up with it. Did a little job here. Zoom out there a bit. Did a nice little job here of sort of labeling the vascular cambium with that little yellow dot and that green dot or oval, if you will, for the cork cambium. Wanted to be able to see that, and the fact that the pith is the all that's left of what's in the middle. So, do a quick little label there. We would refer to all of the xylem as the woody tissue, and the center, which can rot out, really experience quite a nasty fate. There's your pith. And when you look at the bark, you sort of say, well, what really constitutes the bark? It's pretty well from here, from the outside. Give me your pen. Gonna work for me? There we go. All the way down towards the living tissue. So if this is cut, and elephants can be really tough on trees because they'll tear away at this tissue. And if they tear away at that tissue, they're literally killing the sugar conducting tubes, which are kind of on the outside of the plant. Think about it. When you tap into a tree, if this is the side of a maple tree, and you tap into it, you don't tap deeply into the maple tree to get the maple sugar. You just tap just underneath the bark because underneath the bark is where the phloem and the phloem rays are, collections of phloem, if you will. And you tap into that, and you're diverting a little bit of the sugar conducting as it's going from the roots upwards in the plant, keeping the plant fed. Now, if here's the secret: don't girdle the plant. If you cut all the way around the outside and completely destroyed the phloem in a perfect circle, you would kill the tree because you would have cut it off from the top and the bottom of the plant. You can harm a side of the plant as long as you don't harm it all the way around the outside. Okay? Not that you should harm a plant in the first place. Pretty neat when you look at tree rings. This tree here, 1600 years old, has definitely seen a bit of time. We're able to age it because you can, oops, if you go from its pith towards the outside with some careful attention to detail, you can figure out how old this tree is. It was 1600 years old when it died. Estimating the number of years, it goes back to basically 256 BC. That is unbelievable. 1600-year-old tree. If you want to get into a little bit more detail, the heartwood is you've got the heartwood, which is the kind of like the think of it as the older wood, sapwood, which would be the younger kind of xylem. So now you start to get into a little bit more forestry, talking about sort of what the sections of the word and what's most desirable, etc. It's neat the way they did this because you can see the vascular cambium just towards the outside of the sapwood, and then more exterior to the vascular cambium, what you'll see is that layer of phloem right there, right here. And that's why you don't want to girdle a tree, which means go around the outside of it and kill it. If a wire is put around a tree and as it grows, it will cut into the tree in a perfect circle and girdle a tree. That's terrible. You don't want to, that should never happen. Modified stems. Well, let's look at some of the things that we consume, for example. If you look at a strawberry plant, it sends out these little runners like this. And they run across the ground and then they'll asexually reproduce and a new plant will pop up. Okay, so those are referred to as stolons. You can have underground stems like rhizomes, these little guys here. Extended starch storage, if you will. Okay. You can have these little carbohydrate storage elements underground, tubers. Okay. And that's kind of interesting. Really, when we think about it, when I look at something like a tuber, you're really looking at starch storage. Once it's underground, let's just be honest. Let's just call it for what it is. It's really a root at this point, and the plant is just taking, instead of taking its photosynthate and storing it in something like an apple, it's driving it underground and storing it in, let's say, a potato. Okay? Interestingly enough, you can get carbohydrate storage in, if you think about an onion, when you look at it, as you peel it away, it is very different. When you look at something like an onion, that's a little bit different than a tuber. It's not really sort of underground root development, right? It's got papery material as you pull it away, and you sort of think, hey, this is a little bit more like a leaf. Corms are like that, where what you're doing is you're storing carbohydrates in sort of these underground kind of leaves. You see that with your onions and leeks of that variety. Okay, so cross section of a stem. I think we've seen this many a time. Little exercise here. You can press pause. See if you can figure out how old this tree is. Right? What I like, though, not in addition to that, is that this little evidence of the past. This damage right here. Reminds me of a CSI episode I saw where they wanted to find out when someone had died, and it was a pretty interesting circumstance that the tree was nearby and was taking in some of the smoke from the incineration of the poor unfortunate deceased, and they used the tree rings to figure out the time at which the deceased had died. That was quite the extrapolation on science, let me tell you. But nonetheless, when you look at this, it's evidence of what's gone in the past. Forest fire, damage to the outer section of the plant. Nice shot here of how you get woody tissue, right? You can see the vascular cambium contributing to ring after ring. That's all that's about. Bark, if you want to get specific, inner, outer, right? And you can look at the bark that's being laid down Bark is really just the outside. Think of it as the rough protective material, the sort of the tough epidermis of the plant. Now leaves can be pretty interesting. When you look at needles on a cactus, you say, well, what the heck are those? Well, you may not have realized, but in a cactus, the stem is photosynthetic, and the leaves have turned into these spines. They don't even photosynthesize. When you look at tendrils on, when you look at cucumbers or you look at peas and beans and you see them climb, those are modified leaves. They're not so much photosynthetic as structural. So just when you thought you knew something about leaves, Mother Nature comes up with entirely no purpose for them. The Venus flytrap, modified leaf. Always am fascinated by the Venus flytrap. Little pressure sensitive hairs. And if the insect is small enough, it'll be caught in there and this leaf will digest that poor little fly. It doesn't work with beetles. Large critters, they just power their way right out of the Venus flytrap. But I think I need to get one for school. They're so much fun. If we look at the leaf, it should be broken down into what sections it really has. It attaches to the stem right there. So if the stem's here, it attaches with something called the petiole. Now, if you like celery, that's pretty well what you're eating, right? My father loves celery. He puts celery is one of those foods you can eat as much as you want. My father always takes it to the extreme because what he'll do is he'll put Cheez Whiz right up the middle of the petiole, sort of defeating the whole point of uh, that healthy celery where you can eat as much as you want. But hey, when I was younger, I did the same thing. In celery, it just has a reduced blade. There's just not as much sort of foliage there. It's called a node, by the way, where it attaches. So draw it a little bit primitively, but you have to forgive me. I'm writing on an iPad. That's a node right there. And if you had another leaf going on, that's a node. And the distance between the nodes is sometimes called isn't sometimes, it is called the internode. Monocots, dicots, I think I've seen that to death, right? But you should know that dicot leaves have that netted pattern of their veins, right? Where the monocots don't. They're all in parallel. Now, this is when it gets really important. You're looking at a dicot leaf, okay? So a little emphasis here. Dicot leaf. I can't imagine not putting a dicot leaf on any test because it's so structurally wonderful. It says it's the business end of the plant that does photosynthesis. So there's a little cutaway there. Let's label it a little bit more clearly. The xylem and the phloem are in close association in the leaf. The xylem's bringing water. And remember, we need water to make the carbohydrate. Phloem, well, think of it as it's conducting sugar away from this area. Now, these veins, there's xylem and phloem, will spread out because if you think about in a dicot leaf, they net, right? So they're branching, xylem and phloem, like that. And they're referring to parenchyma fibers here. That's just strong. That's just really strong tissue. You don't have to worry about that. But what I do want you to focus in on, you've got an epidermis, just like in skin. Okay, so upper epidermis, lower skin. Okay, and then you've got the palisades, right, from here to here. That is where photosynthesis, that's PS for photosynthesis, is going on like crazy. Okay, some people look at the palisade parenchyma and they don't clue into that. Look at all of the little green bits. Look at all of the chloroplasts. It's full of them because that's its job. If you think about it, light is striking from above, and this is where photosynthesis is going to go on very intensely. Okay, it's closest to the sun, if you will. Let's move down a little bit. We get down into the spongy layer, spongy parenchyma, the spongy layer down here, and it's porous because it's letting in carbon dioxide And it's kind of a loose layer to let in carbon dioxide, and water is flowing out through the guard cells. So it's kind of like a breathing space. And that's important. Some photosynthesis goes on here, but it's the palisade parenchyma that does the lion's share of photosynthesizing. Okay? It's spongy to allow for a gas exchange. So when a plant is releasing waste products from photosynthesis, it will release oxygen here. So I'll write O2. Okay. Plants, you got to realize plants themselves have mitochondria, so they do burn energy. So they will also release some carbon dioxide. Okay. I think people forget that plants produce sugar so that they can burn it. So what do you think plants do at night? Out of the stomata, right, that little hole that I just drew an arrow through, they'll be releasing carbon dioxide because they'll be using the sugar that they have produced for their own growth and development. So, got to kind of realize plants respire producing carbon dioxide waste like we do, but because they photosynthesize, they produce a lot of oxygen as well. So, let's not forget that they need to burn their own fuel. Over here, you have your guard cells, and those cells swell up, right? If it gets quite dry out, those plants will take on moisture and they'll swell, or those guard cells rather, they'll swell and try to cover up the stomatal opening, which is there. I didn't want anything, Siri. Right? So I'll put S for stomate. Let's write the whole thing in. So that's what the guard cells protect, is that hole, the stomate. After a while, if the plant gets too dehydrated, the guard cells, they lose water too, and they can't stop the dehydration that will inevitably happen. So this dicot leaf is pretty interesting. Upper and lower skin, a palisade layer, a spongy layer. It's got these veins with the xylem and the phloem. Xylem bringing the water in, the phloem taking the sugar away, because it's a little solar powered factory here. There is mention, by the way, of a cuticle. And if you rub your finger on a leaf, you'll see that there's a little bit of added wax over top of its epidermis, and that keeps it from becoming dehydrated, which is pretty smart. Okay. Now, this is neat. This is a very different structure. I tell you, when you see the alien eyes like this, you know you are looking at corn. This is a monocot. So, if you look closely, you'll see its vascular bundles, smaller ones here as they go down, all kind of lined up in parallel. And that's pretty typical of corn, right? It's got its xylem here and it's got its phloem. You get the idea. Corn itself is a pretty high heat plant. It likes a lot of, how shall we say, it likes a lot of heat. Um, it does very well photosynthesizing in the heat, that intense heat of the summer. Sure, you need to keep it wet, but I'll tell you, its enzymes work best at high temperature. Notice that it has stomata on the upper surface. And here's a neat little shot of one right here. You can see the stomata. Let's switch colors here. Hit done for a second. Let's go with the brown again. This is a stomata, and there's one guard cell and another guard cell. Kind of looks like a mouth with lips there. I always thought it was kind of fascinating. You don't see the stomata on, when you look at dicot leaves, you don't see them having these stomata on the upper surface. That's really kind of a lower surface thing. So corn, sugarcane, right? Um, because they have parallel veins, they're just higher heat plants. They're into this. So pretty cool stuff. And I think I went on to label this. There's your xylem, and there's your phloem towards the outside. So I was being kind of supportive there. And your epidermis, pointing out your stomata on the lower surface. It's good to know. Let's see here. Let's go to my thumbnails here. Make sure of where I'm at. because I want to get to the very last one. There we go. Okay, so now it's all laid out for you. Um, Remember, a vascular bundle and a vein are the same thing. So if we go like this, I've circled the vein, aka also known as the vascular bundle. Neat little shot of the guard cells. They're photosynthetic too. Um, just a neat view of them. And you can get them to fluoresce. Also another great shot. Now, I would be remiss in talking about plants if I didn't mention photosynthesis. You kind of have to discuss it. So, let's go back. There we go. And look at photosynthesis in general. Now, inside of a plant are these little green bits called chloroplasts, right? They make the sugar using sunlight and these two players right here. And then you've got your mitochondria, which are plentiful throughout, especially the animal kingdom because we don't have any chloroplasts. And they just burn the sugar that's produced. Well, let's see here what happens. If we bring in carbon dioxide and water, and we add a little solar motivation, energy is the ability to do work, right? Well, what happens is we get the production of glucose, and when water is ripped apart, you get the release of excess oxygen. Now that happens, if you want to take a little close up, right? Let's take a peek. And inside of chloroplast, it's pretty neat. They're actually considered to be little photosynthetic organisms. So if we go through the critter's cytoplasm and we get to these little, we kind of get to these little green, this green area, or sorry, rather, the chloroplast is considered to be an organism. Sorry, we'll get through the cytoplasm of the plant. Then let's get to the chloroplast. And the chloroplast itself has these little green pancakes. They're called thylakoids. Now, this whole green region is considered to be trapped photosynthetic bacteria, which is pretty cool. How do we know? Because we can take them out of plants. They have their own DNA. They can be hurt with certain kinds of antibiotics. So having their own genetic code, why are they hanging around in plants? Looks like they got trapped at some point in evolution. Or it's a mutualism. Perhaps it's both. But as water comes in through the plant cell, it enters into this organelle, which we know is actually trapped bacteria, and the water itself needs to be torn asunder. And that's what light is for. Light breaks it up. And this is the part of the reaction you probably weren't aware of. Hydrogen and oxygen are just not going to dissociate without some kind of motivation. Amazingly enough, plants have learned to take solar energy and use it to rip apart water. And that's quite an energetic reaction. Carbon dioxide comes in, it's worked over with some enzymes and the input of energy, and we form, in concert with having some water around, our carbohydrate C6H12O6. If you reduce that, CH2O. And so we get our net reaction. It's just like a recipe. Of course, this one isn't balanced, but we'll let that go for now. Take your carbon dioxide, take your water, light, some minerals, and you get the output of glucose and oxygen. Oxygen, as it turns out, was a toxic product to plants. If you want to know how toxic oxygen is, look what it does to the bodies of cars. It promotes oxidation, right? Rust. But we've learned to take a poison like that, and we can't live without it. And for that matter, plants can't live without it because their mitochondria need it too. And I think people sort of forget that. Plants have mitochondria? Yes, it's the whole reason that they're making sugar. If you want to break it down and keep it real simple, what's amazing about plants is they're kind of like, well, if I was driving in my car and I had a built-in gas station, I could just keep going. If I had chloroplasts, I could feed myself. That's the whole idea. You have to do photosynthesis so that you can respire. And plants are pretty happy that we're respiring because we're producing all that carbon dioxide as well. So a tree if we were, there's an interesting show on TV right now called Under the Dome. Let me extrapolate on that. A bunch of people caught underneath a dome, and it's kind of an interesting, sort of television show. But if you thought about travel into something like space, or, how would you send enough oxygen to keep somebody alive? Well, here's a better idea. Why not send plants? A fair sized tree produces enough oxygen, right? It produces it in excess to produce enough for 4 to 5 humans to respire with. That's pretty impressive. One tree, 4 to 5 humans. So I think I know what I'd be building in my space station: a means of keeping humans fed on long journeys, right? Obviously, plants. For more than just food, I'd be sending it along so that there is an ongoing source of oxygen, because you could never pack enough, right? And humans in turn give back exactly what the plants need, which is carbon dioxide. Okay, I could go on and on, folks. It's a fascinating topic, but this particular presentation, what I wanted to do was focus in on leaves and a little bit more on photosynthesis. We got a little bit of information on stems as well, modified leaves, modified stems, roots, etc. But it's a pretty good topic. Okay, folks, Mr. B out. Have a good one.